mitotic cell division is a process where the mother cell is divided into two daughter cells having the same amount of genetical material. But the mitotic cell division is further divided into two parts, that is asymmetric cell division and symmetric cell division. Asymmetric cell division means the mother cell is divided into two daughter cells, but one cell is equal in the size and shape of the mother cell, where other one is quite smaller. This type of cell division is developed in the stem cells, the asymmetric cell division. What happened? We know that when the stem cell is divided and produce the two daughter cell, one of the two daughter cell having the own stem cell property, where the another one is the differentiated special type of cells. But the symmetric cell division is a process where the mother cells and the two daughter cell are same size, shape, exact. The two daughter cell are same size and shape. So this is all about the asymmetric cell division and symmetric cell division. In the mitotic cell division, we understand the two phases, that are the I phase or interphase and M phase. I phase is responsible for the cell growth and synthesis phase, where the M phase is also known as the nuclear material segregation phase. And after the M phase, there are two possibilities can be happen. One, the cell will either enter into the G0, G0 or quiescent phase and perform its normal housekeeping function, or overlap the G0 phase and enter another G1 phase and continue another round of cell division. But the question is, why should a cell enter into a cell cycle? How it has been possible? Cell doesn't have its own brain to divide, so there must be some kind of activator required that are responsible to either suppress the cell into the G0 phase or may push the cell into the G1 phase for another round of cell division. Okay, so here I am drawing a big cell, just not big cell, actually I am zooming a cell. Okay, so this is the plasma membrane of the cell. This is the plasma membrane of the cell. Okay, and inside the cell, this is a nucleus. This is a nucleus. Here in the nucleus, we know the chromosomes are present, but I am actually what happened? I am actually zooming the chromosome and produce a DNA molecule. There are no difference between the chromosome and DNA. DNA or chromosome are the same structure. DNA are the simplified structure of the chromosome. When DNA are compact in extremely, then it will become known as the chromosome or chromatid. So what happened? The DNA consisting a gene. The first gene, whatever the gene is, think about what happened. The first gene is responsible to produce through the, as if you show our video based on the cell biology first classes, the introduction of cell, you can understand that how from a gene a protein is produced from nucleus to ribosome to endoplasmic reticulum to Golgi body. So think about it. The first gene is responsible to produce a protein. The first gene is responsible to produce a protein, and that protein they throw out of the cell. The first gene is produce a protein, and the protein is, they throw the protein from outside the cell. Now in response to that, a second gene, maybe in the same chromosome, same DNA or different DNA, the second gene is responsible to produce a protein. Okay, this kind of protein, and this protein and became situated here. This protein is situated here. So this protein is actually the receptor for the previous protein. Now the previous protein came here and entered into the receptor protein. Then a conformational change happened, and in the same time a third gene is responsible to produce another protein. Think about this kind of protein. So this protein will intracellularly bind to the receptor intracellularly, and this produce a complex. So now what are the name of this protein? The first gene responsible to produce a protein, and they throw the protein outside, and that protein became bind with the receptor. This is known as a growth protein. So the growth protein bind with a receptor that are made up by the second gene is known as the simply growth receptor. Okay, and the third protein that are responsible to bind intracellularly with the receptor and produce a complex, it is actually a transcription activator type of protein. Actually this complex is responsible to mediate the signal transduction process, and this signal transduction process send a signal and activate another DNA or gene that are responsible to activate a protein known as cyclin. Okay, another protein is known as the kinase. Now the name kinase stands for an enzyme. When from biochemistry we can say that when kinase name is involved, that means there must be some kind of ATP relationship. So kinase is an enzyme that actually phosphorylate the serine or threonine residue of its target protein. The serine or threonine residue actually kinase phosphorylate and activate or inactivate several kind of proteins. Okay, now what are the cyclin? But the problem with the kinase is the kinase can never work alone. They require a friend for their functions. They depend on something, and this protein is known as the cyclin. The cyclin, the name cyclin stand for because in the entire cell cycle we can see that the cyclin level rises and lower in the different phase, because each phase is containing its own cyclin and CDK. Think about it. If A cyclin is responsible for the G1 phase, so it became inhibited in the S phase. Okay, so X cyclin rises in the S phase and became inhibited in the G2 phase. Y cyclin is responsible for the G2 phase. It became inhibited in the pro phase. So each cyclin is responsible for optimizing the function of kinase. So such kinase is known as CDK, cyclin dependent kinase. They are together, the complex of the cyclin and CDK is known as cyclin CDK, cyclin bound with the cyclin dependent kinase. This is all about the function. These are the signal by which a cell are responsible for the cell division. If experimentally we use the blocker, or if we mutated the genes responsible to produce the cyclin kinase, or we block the gene for the growth factor protein or growth factor receptor, what happened ultimately? The cell will remain in the G0 phase. They will never enter into the G1 phase. So actually what happened? The cancer or apoptosis, both are related, and the two consequences of the cell cycle, the cancer and apoptosis. Cell cycle is a normal thing. Cancer and apoptosis are the two consequences of the cell cycle. What happened? They are just the opposite of each other. In our last lecture we talked about that. If the G2 phase protein is being damaged, so mutation cannot be rechecked and it will produce the cancer. But if the G2 protein will be overexpressed, they will clear the, or normally expressed, they can bypass the mutation and clear the mutation. The same thing, think about it. If a cell having some kind of damage, so it is very important to remain in the G0 phase until they die. Rather, they will enter into another cell cycle and produce more and more wrong type of cells. Okay, so this the factor that are responsible to inhibit the growth protein, growth receptor, inhibit the cyclin and kinase genes are responsible to promote the apoptosis. If we use the blocker, or if naturally blocker is used against such inhibitor, okay, these are called double negative gate. A is responsible to inhibit the B. Now if we inhibit the A by a X, then B will extremely produce. So think about the same thing. Think about it, a cell is undergoing through the damage condition. Okay, now it is beneficial, cell must be remain in the G0 phase, not entering into another cell cycle, because if it is entered, it will be produce more and more damaged cell. So there must be a good inhibitor that inhibit the growth protein, growth receptor and cyclin and kinases. So the cell will never enter into the G1 phase. But if we use the blocker, or if naturally that inhibitor has been blocked, then what happens? Growth factor, growth receptor, kinase and cyclin will produce, and cell will enter into the cell cycle, and more will your damaged cells are produced, leading towards the cancer. That's why in our cancer lecture we will understand cancer is not developed by the single mutation. It is developed by the so many mutation in a cooperative way. Cancer will never develop. Cancer may develop into the G2 phase if the G2 phase protein is being inhibited. Cancer may develop into the this G0 phase if the G0 phase has been overexpressed. Cancer may develop if the S phase protein, means the DNA replication protein will be not function perfectly. These are the consequences or the reasons behind this the cancer will produce. The cancer will never develop in a single mutation, but develop by a cooperative mutations. So this is all about the cyclin and CDK. And if we understand in a graphical representation, if we show in a graphical representation, we can this think about it. This is the S phase and this is the G2 phase, and this is the G1S cyclin we are talking about. So G1S cyclin amount is very high in the G1 phase, S phase, but it will become low in the end of the G2 phase. Okay, so these all are happen in the cell cycle. Cyclin came from the cyclic process, because in the different stage its amount became rises and low, getting lower. Now the cyclin, the first cyclin is your first cyclin has been discovered, not G1S and G2 cyclin. The first cyclin is discovered in the M phase cyclin, and at that time no one's understand that it is a cyclin. They talk about that this factor is responsible for the M phase, and then known as the MPF, mitosis promoting factor. Later it will be understand that actually these are the cyclin that are responsible for the mitotic cell division. And in the later study or discovery, the G1S and G2 cycle became produced, and the name of the cyclin is actually naming of the cyclin is actually based on that discover era. Okay, so what happened? I'm telling you about the MPF. The good model for the cell cycle is actually yeast. Yeast is a very good model for the study of the cell cycle, because you don't need to knock out the gene. The yeast is the proteins of the yeast are extremely temperature sensitive. So if you wanted to know what happened in the mutation, you don't need to knock out the gene. you just alter the situation. Take an yeast which is a normal temperature, now put into a high temperature or in a very low temperature, the protein will be non-functional. So you can easily understand the mutation. So there are two type of yeast are required for cell cycle study. One is the budding yeast and another is the fission yeast. The fission means means the one cell is divided into two in both cases, but budding technology and fission technology. The fission is very similar like in the eukaryotic cell cycle. So these are the two. Now the MPF of the budding yeast and fission is CDC2 and CDC28 they call. And in case of the eukaryotic cell cycle, the MPF is known as the cyclin B1, CDK1 or 2. These are the responsible for the MPF or M phase cycle. So this is all about the cell cycle. Why cell cycle happened? How, what is the symmetric asymmetric cell division? How the cell growth factor is responsible to initiate a cell cycle? What is the cycling? What is the CDK? How their regulation has been altered during the different phases of the cell cycle. This is all about the cell cycle. Now we are in the next lecture we will enter into the brief discussion on the each step of the cell cycle.